topic, I want to mention a couple of things. First off, you probably noticed that there are people in this room that you don't recognize. Because of the holiday coming up this week, I told my Wednesday class to come on Monday, which actually turns out to be pretty good because the dean this morning emailed out to say that the school's probably going to cancel Wednesday afternoon classes. Now, next, after the dean says something like that, and I meant Wednesday afternoon classes for Thanksgiving. The way things work at this school basically is somebody from administration says, okay, here's what we're doing. And then this backlash of response saying, you can't do that on the Monday before you're going to make a change. It's really a bad idea to say today we're going to change the schedule. It's not like they didn't know Thanksgiving was coming. They've probably known that for a while. And it's not like we don't understand that people don't always come to Wednesday afternoon classes. And in fact, that's why I originally suggested to my Wednesday afternoon classes to come today instead. The problem is that making some change like that at this point in the semester can really mess things up. And I'm sure that there are some of my colleagues that are going to be quite irate about it. But I actually already kind of anticipated it, so I set it up this way. So that's why you're. I am recording what I'm doing today, capturing the lecture. So people that aren't here, because there were a couple of people that had work issues and they couldn't come in today, make it in on Wednesday, Monday instead of Wednesday. And then there's a profound lack of Monday class people here anyways, and it is Monday. So I am capturing this for the sake of people that can't be here today. So I'm actually in last week's folder right now because I just wanted to point out that I am in the process of putting the recordings up. I have 2 more to do, one on gas exchange and one on regulation and health things, which I kind of hope to get done tomorrow morning, but usually when I say those things, something comes up and I don't get to finish it. That's my hope. But they will definitely be up there sometime this week. So, and then I have to get to the digestive system, which we're doing today anyways. But that's all coming up. So we are in the digestive system today. What I want to do is to actually, did I talk about this with Monday class last week? That I think I did on Wednesday, but I'm getting behind on the putting the stuff up on the with the presentation stuff up. So I'm really going to really for the rest of the semester sort of default back to the way that I used to cover the material in this class, which was in the 3 some odd hours that we have each week. I'll cover what I can and highlight some important stuff and leave some things to what I expect people can get on their own out of the material. And I'll talk about, you know, like, I'll point some things out and I'll say, I'm not going to talk about such and such because you can get that out of the reading more directly or something like that. So I am going to cover things that way. Oh, I remember I did this whole spiel Wednesday night. Anyways, I will today do what I've always done for the digestive system in the past, and then I'll do the same for the urinary system and the reproductive system for the other parts of this semester. The online content that I've been putting up has, to some extent, been more of an added feature for this semester. And especially now that I'm just across the board saying I'm not switching the way that I'm teaching the class for this semester, kind of recognizes the point that I'm behind on getting those things up. And I want to give you the material like I've given it to students in the past. I will be getting those things up. My intention is to get all of those up into the system this semester. So that I have them ready for next semester, because I will be a little bit busy next semester and don't have as much time to put stuff up. So they will be there, they just won't be there in as timely a fashion as I was hoping. The due dates for material are as they are. You are aware, or you should be aware because I've said it several times and it's in the syllabus, that the consequence to completing things late is that it pops up in my needs grading section. And I know when you're doing things late, and if you need to do an assignment that's dependent on a previous assignment, you can't go on to that until I check off things in the needs grading section. Which means that if you do part 1, 2, or 3 late, you won't be able to go on to do part 2, 3, or 4 based on that until I check things off, which can be an inconvenience, which you wouldn't have if you do things based on the due date. But other than that, the consequences are minimal at best, practically non-existent. There's no effect on your grade to do things later or anything like that. So if you'd rather have that online content available when you're working on chapter assignments, you're welcome to decide to hold off on doing those things till later so that you have the added material available to answer questions. But it will not be up in a timely fashion for you to hit the due dates on that. Now, really, for any set of assignments in a chapter, part 1, 2, 3, 4 assignments, the part 1 and 2 assignments are really about the reading. So what I place online is really more for the part 3 and 4 assignments. So if you're saying, I'm going to hold off on doing the digestive system stuff until he has the digestive system online content up, that only really applies to part 3 and 4. You should get part 1 and 2 done on time. And then you have part 2 done on time and you're waiting to do part 3 and 4 once I get things up. Then the only inconvenience you're going to be dealing with is you'll do part 3, it'll be late, and you have to wait for me to check off part 4. So that's probably a good way to kind of deal with the material if you would like to rely on the added material I'm putting up. It just won't be up in a timely fashion, but it really only affects part 3 and 4. You should be able to do parts 1 and 2 based on reading. That's the way that those assignments were always designed. Okay. So, what I want to do today in the first half is to kind of address the overall conceptual anatomy of the system, which is to some extent this section here on overview of digestive system. I'll talk about the processes and regulation stuff in this section, but I'll mention it briefly. But there's not a whole lot in that doesn't come up in the second half. But in the first half, besides just mentioning these things about the overview of the anatomy of the system, I also want to talk a little bit more about some material that's sort of in this last part of the chapter here. It says chemical digestion and some other stuff. And I want to go into that a little bit more to illustrate some, again, overview type concepts on the system. And then in the second half, I want to address the structures and function of the digestive tract in more detail. So in the book, they have a section on the mouth, pharynx, and esophagus, which is really about chewing and eating, chewing and swallowing. And then I want to talk about the stomach, and that's going to get the most amount of time just because of the way things work out. And then I'll address some things about the small intestine and very cursorily mention some things about the large intestine. And then this section up here is essentially about the pancreas, liver, and gallbladder. So I'll address that. And the more directed structure and function of the digestive tract, that's sort of the goal of the second half. So in the overview of the system part, it enumerates the digestive organs. I'm not going to, well, I am going to list them real quick because I want to define two aspects of, or two types of digestive organs. Got to keep in mind that I'm doing this with the lecture capture, so I want to do stuff on the screen as much as possible. So here's just a picture showing the different digestive organs. And digestive organs fall into two groups. I'm going to type out this list here, and I'm going to add some things in between stuff. So if you're writing this down, I suggest that if you want to keep up and take your own notes as I'm saying, you're fine. That's fine. But I'm going to have sort of the final version of this list after I describe what these things are. So the digestive tract is all of the organs that food actually passes through. And it's also known as the alimentary canal, which I think is a term that the book tends to use. Also, it's the gastrointestinal or GI tract. And if you just think about where the food goes, obviously you put it into your mouth, so it starts with the oral cavity and then goes into the throat or pharynx, then into the esophagus, and then to the stomach, the small intestine, and the large intestine, which is also known as the colon. There's just fine distinction between what is the large intestine, what is the colon, but they're basically overlapping concepts. So there are those. The accessory digestive organs are those things that aid with the function of the digestive tract, most broadly speaking. Most sources will talk specifically about organs that secrete digestive juices into the digestive tract. So that starts with the salivary glands, which secrete saliva into the oral cavity. There are three of those. There's the parotid gland, the sublingual gland, and the submandibular gland. The first one, the parotid gland, it's just its special name. The other two, their name tells you where they are. The parotid gland is basically near the temporomandibular joint, just anterior to the ear. And then sublingual means beneath the tongue. Submandibular means beneath the mandible or the jawbone, which of course there's nothing technically below that, but it's in the bottom, the soft tissue at the bottom of your mandible. And then there's the pancreas, which is responsible for making most of the enzymes that are used in the small intestine, and the liver, which is responsible for making bile, and I'll explain what bile is later on. And I like to include the gallbladder alongside the liver, because all the gallbladder does is it stores the bile that the liver makes until it's needed for digesting a particular meal. And hopefully that'll be obvious when I get to explaining that. So those are the organs that make up the tract. I'll go back to this list in a second. I just want to remind you of this picture here in the book, which you can use this list and find the various structures there. I think most of them people are probably already familiar with. And I'm going to, other than just listing these out into the two different groups, I'm going to leave it to you to look at where they are and what they look like. We will talk about the structures in the digestive tract, and then the pancreas, liver, and gallbladder in some detail in the second half today. But other than that, I'll leave it to you. Now, the other aspect of the digestive system anatomy here in the overview section is depicted in this picture here, which let me get a larger image of. This is just showing the layers of tissue that make up any digestive organ. There are 4 layers, and if you look at the labeling here, there are 4 terms that are bold-faced: mucosa, submucosa, muscularis, and serosum. Those are the 4 layers. The mucosa is the mucous membrane that lines the inside of the tract. It's what's directly adjacent to the food as it's moving through. It contains an epithelium, and then its associated connective tissue called the lamina propria, which is just standard for any mucous membrane. You always call that connective tissue lamina propria. It is an areolar connective tissue, but it has a special name. And then there's a very thin very thin associated layer of smooth muscle called the muscularis mucosae, not to be confused with the muscularis, which is a separate layer altogether that I'll get to in a second. That's probably like 2 cells thick, very, very thin layer of muscle tissue there in the mucosa. Now, the epithelium varies depending on where we look at the digestive tract. The oral cavity, the pharynx, and the esophagus are all lined with a stratified squamous epithelium. The stomach and the intestines, both small and large, are lined with a simple columnar epithelium. So if we go back to this list here, we can put a line between those two. Everything above that has a stratified squamous epithelium. Everything below that line is simple columnar. Okay. Can anybody tell me what anatomical structure corresponds with that division there? It's not a digestive tract organ, but what else is going to be separating those organs into those 2 groups? Strictly anatomical structure. Any ideas? It was a big player last week. Right, the diaphragm. Okay. So that's to say that the stomach and the intestines are in the abdominopelvic cavity. The esophagus is in the thoracic cavity, and then the pharynx and oral cavity are in the skull, head and neck. But that line, again, which essentially is the diaphragm, is also a line that separates the epithelium lining the digestive tract. Personally, from an anatomical point of view, I think that this is the best definition of upper gastrointestinal tract versus lower gastrointestinal tract. Now, I'm actually setting something up by saying GI tract rather than digestive tract, but it's just an alternate name for that, and I'll come back to that a little later on. But that's the epithelium in the mucosa. The submucosa is just connective tissue that's deep to the mucosa. It has some glands that extend outside of the mucosa. So if you look at the mucosa layer here in the very center, there are some glands that are part of the mucosa, and they have one gland depicted here that extends past the mucosal layer, and in this part of the picture here, it's just kind of floating in no man's land. They don't have any tissue surrounding it. But here's another one that is, in fact, in the submucosa, or another one drawn here that's not labeled. So the deepest part of this gland here would be in the submucosa, just the way they've set up this picture, that tissue isn't represented surrounding it, but that's in fact the case. Okay. The submucosa has a layer, not a layer, but a region of nervous tissue called the submucosal plexus. The word plexus kind of means network. So it's a network of neurons and axons and those sorts of things in that layer. The next layer is the muscularis, which is made up of two sizable layers of smooth muscle compared to the muscularis mucosae. One layer are fibers that go around the tube that we're looking at, and so they're called the circular muscle. And then another layer are fibers that run the length of the organ, and so those are longitudinal fibers. The intestines and the esophagus look like what we're talking about here. Now, technically, this picture suggests the small intestine, but the large intestine, the small intestine, the esophagus all have a muscularis as depicted here. Now, I left the stomach out that's in between those because the stomach has an extra layer of muscle, has a third layer that's called the oblique layer, or the oblique muscle, which just, the word oblique means at an angle, so those are fibers that run at an angle to the circular or longitudinal fibers. And when we get to talking about what the stomach does, it'll be obvious why it needs additional muscle in it. As we get into the pharynx and the oral cavity, muscularis is kind of hard to describe. There are muscles in the wall of the pharynx and in the wall of the oral cavity, but oftentimes they're skeletal muscle fibers that are part of distinct groupings that we have specific names for. For instance, in the wall of the oral cavity, we have the buccinator, or buccinator, however you pronounce it. It's in the wall of the cheek. It would be a good example of muscularis for the oral cavity, but it's not smooth muscle and it's not arranged exactly like these. It's a muscle that goes sort of from the joint at the jaw to the corner of the mouth, and when it contracts, it basically helps to push food in between the cheeks, in between the teeth for chewing. But that's probably the simplest example, something that's most like maybe a longitudinal muscle for the oral cavity. But we could also say that the tongue is muscle tissue that's in the oral cavity. And obviously it's very different, it has a bunch of other structural elements to it than simple a couple layers of tissue. And then in the pharynx also there are specific muscles there that don't exactly fit into a definition of the muscularis. But that's because up in that end of the digestive tract, we have more voluntary control over what we're doing, or at least reflexive control. And so we have different types of muscles there. This serosa is a serous membrane. Now, we've already talked about two serous membranes in this course so far this semester. There was the pericardium, which is around the heart, and the pleura, which is around the lungs. The serosa, that's part of the digestive tract, has a special name for it also, which is the peritoneum. If you've ever heard of an IP injection, that means intraperitoneally, and that's an injection into the space of the abdominal cavity. I believe that's the route that insulin is usually injected, although I might be wrong about that. I'm used to IP injections because they were very common in research with rats. You just pull the skin of the belly up the rat, and you can go in and get an injection into that the abdominal cavity very easily that way. But this is not rat anatomy. Now that serosa, the peritoneum, covers the intestines and the stomach, and to some degree the liver, the pancreas, and the gallbladder, which means it's only covering the digestive organs in the abdominal cavity, abdominal pelvic cavity. We also call that cavity the peritoneal cavity because it's lined with the peritoneum, just like the pericardium lines the mediastinum or the pleura lines the rib cage. It's lining that space there. There is no serosa for the esophagus, pharynx, or oral cavity. The esophagus, instead of having a membrane on its outside, instead just has connective tissue anchoring it in place in the thoracic cavity, much like blood vessels have loose connective tissue in their outermost layer. And then the pharynx and the oral cavity, the thought of an outer covering for those is unusual just because they're really incorporated into the rest of the structure of the head and neck. I suppose you could say that the skin in the cheeks acts as some of the outside covering of the oral cavity, but it's just a weird way to think about it. So the serosa we tend to think of as only being inferior to the diaphragm for those organs there. So going back to this list here, I just want to point out we have made a split between the oral cavity, pharynx, and esophagus, and the stomach and intestines. The diaphragm is a physical barrier between those two cavities. It's the demarcation line between where the mucosa is lined with stratified squamous epithelium superior to the diaphragm, or simple columnar epithelium inferior to the diaphragm. Also, now we just covered that the organs inferior to the diaphragm are covered with the serosa, the peritoneum, which also includes the pancreas, liver, and gallbladder down here at the bottom. But those are not, I guess, tract organs. This dividing line sets up the upper GI and lower GI. Again, I'm using GI on purpose there. When you hear of an upper GI, especially in a clinical setting, what does that usually suggest? Does anybody know what an upper GI refers to? Or for that matter, a lower GI, although that's a less common term. What?
Esophagus.
Say that again?
Like your esophagus.
But what about your esophagus? They want to look at it. It's a test to look at the upper gastrointestinal tract. But it looks at more than just the esophagus. What is it also going to look at? Upper GI. This is called an upper GI endoscopy. If you've not heard that before.
So it would be your oral cavity, pharynx, and esophagus.
So it's going to go in, it's going to see the oral cavity, it's going to see the pharynx, it's going to see the esophagus. But what else does it see? Has anybody ever had an upper GI done? Really? Okay. Usually there's one or two people. It'll also go down to the stomach and maybe even to the very beginning of the small intestines. So it's usually looking for evidence of, if somebody has bad heartburn, looking for evidence of damage to the esophageal lining or something like that. But if somebody has bad heartburn, they also want to look in the stomach to be sure that there isn't damage to the lining in the stomach in the form of ulcers or something like that. And potentially even down into the duodenum, which is the first part of the small intestine. So clinically speaking, an upper GI, meaning an upper GI endoscopy, can actually include the stomach as one of the things that it's visualizing, as well as getting into the small intestine. So if there's such thing as an upper GI endoscopy, there's also such thing as a lower GI endoscopy. Does anybody know what the more common name for that would be? Right, that's a colonoscopy, which is only to look at the large intestine, hence colonoscopy. You would not want to pass that camera from the large intestine into the small intestine. There's a valve or sphincter between the two intestines that remains closed and only allows things to move from the small intestine to the large intestine, meaning food stuff. There are bacteria that live in your large intestine on purpose. They have a very important role to play there. But we don't want them migrating out of the large intestine into the small intestine. So there's a valve there to make sure that they can't come back. But if you take a camera and push that camera through the valve, you'd probably damage the valve, and it would no longer keep things from the large intestine from getting back into the small intestine. So a lower GI endoscopy is really only looking at the large intestine. An upper GI endoscopy is a better way to get at looking at all of these other things. From a clinical perspective, an upper GI might refer to looking at the stomach. Okay? From an anatomical perspective, the stomach is in the lower digestive tract or lower GI from an anatomical perspective. Just there are different ways of defining what things are. If you go online and you do a Google search saying what is upper GI or what is lower GI or digestive tract, you'll probably get the classical explanation for that, which is based on embryology. The tissue that makes the upper digestive tract organs and the stomach and even a little bit of the small intestine comes from one part of the embryo, and the large intestine and the rest of the small intestines comes from a different part of the embryo. So the classical embryological picture of upper versus lower digestive tract is not the same as what we see here. I'm actually kind of surprised that this very clear demarcation between the esophagus and the stomach isn't just generally accepted as the dividing line, because there's three things that this line speaks of. One, it's the location of the diaphragm. Two, below this line is simple columnar epithelium. Above this line is stratified squamous epithelium in the mucosa. And three, below this line, we have that serosa, the peritoneum. We do not have peritoneum above that. So from an anatomical point of view, there's a very, very clear distinction between upper and lower. But clinically, an upper GI endoscopy is going to look at the stomach too, and possibly some of the small intestine. Just, it's, what's the word I'm looking for? Well, it's easier to do that. You can't put a camera in through the colon and look at the stomach. Okay? Besides the problem with the valve between the large and small intestine, you can't possibly get a camera to traverse through the entire small intestine to find every little nook and cranny and hole and get back to the stomach reliably and without causing damage. So it's just more feasible to get a camera into the stomach through the oral cavity. Clinically, the stomach fits in with the upper GI as far as it's being looked at. So that's what I wanted to say. That's the overview of the system, okay? Which is, the book starts with what are the organs of the digestive system, and then the tissue structure of a digestive organ. And the picture here is showing us the small intestine, which is sort of the general example of that. But I applied how there are some differences with other structures, but that's basically what it is. The book then I didn't mention, or I did mention the submucosal plexus. I meant to, when I was talking about the muscularis, to mention the myenteric plexus, which is nervous tissue that's sort of embedded within the muscularis. The nervous tissue that makes up these two plexuses, the myenteric plexus and submucosal plexus, collectively is known as the enteric nervous system, which is nervous tissue on its own that has its own set of functions, and it helps to regulate digestive function. But it's also a component of the autonomic nervous system. For the autonomic nervous system, what do the two different divisions control as far as digestive system is concerned?
I'm sorry, what was the question?
What do the two autonomic nervous system divisions control in the digestive system?
The parasympathetic increases the digestive.
Right. The parasympathetic system is associated largely with increasing digestive activity. And in fact, when we refer to the parasympathetic function, we sometimes use the eponym rest and digest. So you digest food. And possibly Thursday afternoon, you're all going to be doing a really serious job of that. what does the sympathetic system do to the digestive system? Slows it down. Okay. Largely because when you're in fight or flight mode, digestion doesn't do any good. One, you're not going to make use of any nutrients that you absorb from the digestive tract immediately. It needs a little bit of time to get through there. And two, instead of sending blood to the digestive tract to absorb those nutrients, you'd rather send it to skeletal muscle, where it can provide oxygen for those sorts of things. And with that, the sympathetic system is also very important in making sphincter muscles contract. So there's a sphincter between the esophagus and the stomach, and between the stomach and the small intestine, and between the small intestine and the large intestine, which is that valve I mentioned a little ago. Those muscles, as well as sphincters at either end, are under sympathetic control because you want to stop food from moving through the system while you're not using it. And so the nervous tissue we see here is the sympathetic and parasympathetic structures associated with controlling the digestive system. Now, in the two plexuses here, we have a number of neurons, which we could consider in ganglia, although technically ganglion is not the right word for it. But which ganglia would they be that are here in the wall of the organ, as far as the autonomic system is concerned? Right, those are the terminal ganglia of the parasympathetic system. So any neurons that you see here would not be part of the sympathetic system. The sympathetic system is controlled by the chain ganglia, or more likely for the intestines, the prevertebral ganglia. And the axons from those ganglia end here and release their norepinephrine to have an effect on the organs. Now, I want to just pull up the Prezi, oh, that's it, because I want to use this to talk about the next thing. Give it a second load. So I just like this cartoon, sort of the splash page cartoon showing, input of a cheeseburger, add saliva to it, mix it up a little bit, send it down. Oh, sorry, this is supposed to represent the stomach, right? Yeah, and then it goes into the small intestine where the pancreas or the liver or the gallbladder supply additional stuff to it, and we get out of it what we can. We absorb nutrients, and then eventually it goes on into the large intestine where we get rid of all the waste. And then this trough that we're collecting the nutrients in represents our bloodstream. And waste from the bloodstream is pulled out in the kidneys, and we get rid of that in our liquid waste. So this also shows a little suggestion of how the digestive system and urinary systems interact in a very cartoon format. But what I really want to get to is look at this stuff here. This list is sort of the major functions that we associate with the digestive system. Actually, jump out of this for a second. The next section in the book is digestive system processes and regulation, some of which gets at the sort of overview of the autonomic control that I just mentioned. This mention just gives a list of all of the different things that's happening throughout the digestive tract. And this is one introduction of a picture of what it means to move food through the digestive tract. So that sort of goes with this list here to some degree. I just want to spend some time talking about digestion in a little bit more detail. And you might not be able to read it from where you are, but there are two words here, and I want to zoom in on these and talk about what digestion is a little bit further. What else is listed here, I'm just going to mention real quick. Propulsion obviously needs to move things along, so The digestive tract especially is pushing food through the system from the beginning to the end so that we can digest it, which I'm going to talk about, and absorb material out of it. Secretion is referring to glands that are secreting digestive juices, as well as some of the mucosa has cells that secrete things directly into the digestive tract. And then at the end, of course, we're going to eliminate all of the indigestible material. Now, I want to talk a little bit about what digestion is. And there's two aspects of what is happening to really digest our food. There's mechanical and chemical aspects that drive that digestion. Mechanically, we're talking about breaking food apart. So the most obvious example of that is chewing. When you put food in your mouth, you're going to use your teeth to break it down into smaller and smaller pieces. And then liquefaction, I suppose, is kind of a broad term depending on where we're looking, but we're always trying to dissolve the food into a liquid medium. So we're mixing the food with saliva in our mouth, but actually with chewing and saliva mixing with it, we might get it to a kind of pasty consistency or something like that. And that pasty consistency food will be swallowed when it gets down to the stomach. Liquefaction is really the main part of what the stomach's doing, which is to take the at least semi-solid food stuff that's been swallowed and completely dissolve it into a liquid environment. And then that liquid is going to continue on through the intestines as we absorb all that we can out of it. And finally, we absorb all of the water out of it, and what's left over, the indigestible material, is at least back to a sort of semi-solid consistency, and that's what we get rid of in defecation. And then propulsion is actually the next thing on the list after digestion, the big list I just showed you, but it's also really a form of mechanical digestion. We're dealing with semi-solid, pasty, whatever you want to call it, food stuff, or even liquid, dissolved food stuff. And as you move that stuff through a tube, it's necessarily going to mix it up a little bit. And that mixing coming from just the movement of the food will also help sort of mechanically rearrange the components of the food so that we can break it down a little bit. The chemical digestion aspect can be addressed looking at these three different ideas. First, solvation means to dissolve the food into a liquid. Now, that's not liquefaction from above, but rather talking about those nutrients in our food that all we need to do is to get them from their solid form to a dissolved form. And the best example of this is table salt. So if you put sodium chloride on your food in crystal form, as soon as it goes into your mouth, it'll dissolve into your saliva. And then you have sodium ions and chloride ions. And we can't do anything more to those. Those are at their basic level for us to use in our body, and we'll eventually absorb them. So sometimes dissolving the food is all that's necessary. For those molecules that need more work to break down to their constituent parts, we have enzymes that can break them down. And these are proteins that are secreted from a lot of our digestive glands that target specific types of molecules and break them down. I want to talk about those in a little bit more detail, which is what I'm going to move on to next from this part of the presentation. But then the other aspect of chemical digestion is emulsification. Now, this isn't really chemical digestion in that we're breaking the chemicals apart, but emulsification is necessary for chemical digestion to take place. And you might not recognize the word emulsification, but you've probably dealt with it plenty in your life. Who eats salad? You probably don't eat salad dry. It's not that interesting by itself. So what do you put on it? What kind of dressings do you eat? Ranch, Italian, balsamic. So wait till the ranch a little bit. The Italian or the balsamic are essentially oil and vinegar mixtures. Okay? And what do oil and vinegar not do? Mix. Okay? If you have a bottle of Italian or some sort of vinaigrette dressing, it's probably going to be separated into two phases, an oil phase and a water phase. The vinegar is the water, and then the oil phase. To get them to mix together, that's emulsification. Emulsification is forcing hydrophobic lipids to mix with hydrophilic water solutions. So how do you mix oil and vinegar in the bottle? Okay, you just shake it up. That's simple emulsification. It breaks the lipids down, not breaking it down, but separates the lipids into small oil droplets, and then those get suspended within the water-based solution of the vinegar, and then when you pour it out on your salad, you get a nice mixture of oil and vinegar there. Okay. Now, onto ranch. You don't have to do that with ranch, because ranch or blue cheese or Russian or whatever dressings are all based on pre-emulsification. What's the basis of a ranch dressing? It's creamy, but it's not cream in the sense of what you skim off of milk. Anybody know it's the base of ranch dressing? What?
Is it mayonnaise?
Mayonnaise, right. So if you go to the store and you go to the dressing aisle, you're going to see a whole shelf full of mayonnaise jars. Mayonnaise is salad dressing. We don't use it that way. We now use it as a condiment on our sandwiches or something like that. But it is the basis for that. What is mayonnaise? Anybody made mayonnaise before? Anybody watched it being made?
Eggs and something else.
Yeah, eggs and something else. What's the something else? Anybody watch like the Food Channel or anything? They make it all the time there. Eggs and oil. A little olive oil and egg, mix it up, you got mayonnaise. It's pretty bland mayonnaise at that point, but you add some stuff to it to make it taste better. Actually, when you do that on the Food Channel, they call it aioli, but that's essentially mayonnaise. The reason why mayonnaise is a homogeneous mixture of this creamy kind of stuff, which is the basis of ranch salad dressing, is because it's already emulsified. There's a substance in egg that will emulsify lipids and mix them in with water-based components so that you get a homogeneous mixture. I happen to know that stuff is called lecithin, because I actually use lecithin in my kitchen a little bit. We have a dog who has epilepsy, and so it's got to have sort of special made food. And so one of the things we have to do is make food for it every couple of weeks, a few weeks, whatever. And so there's an oil component to that food as well as some more water-based stuff. And so we have to put lecithin in it to emulsify, which I could probably just use an egg, but then that introduces all the other things in the eggs, which I'm sure the dog would love. But the recipe I have calls for lecithin. And so I go to the natural food store and I buy a little pack of lecithin. The interesting thing that I've learned about that for lecithin, since it comes from an egg, is that the lecithin that we use usually for emulsification comes from an egg, is that vegans obviously can't use it. So when I buy lecithin at the store, I'm buying, I used to buy a soy lecithin, but then of course, when Health Food Nuts got all freaked out about GMOs, I stopped using that because soy lecithin is a GMO. So sunflower seeds apparently have a good source of lecithin. So I get sunflower lecithin for the dog food. But all it does is it gets in there and it helps the oil that I mix in emulsifying with everything else. Okay. So that's what emulsification is, but that's not what it is for our digestive system. The reason I'm talking about it so extensively is because you've probably had experience with salad dressing, whether it's oil and vinegar or a mayonnaise-based sort of thing. And that's illustrates what emulsification is. But in our digestive system, what emulsification is about is when you eat lipid-rich foods, you have to be able to break those lipids apart because everything's happening in a water-based environment. Okay, remember I just said liquefaction is largely in the stomach where the food gets dissolved into a water environment. Lipids aren't going to do that. So when lipids are in a water environment, they're usually going to clump together. So if you eat a big cheese pizza, all of the milk fats from the cheese, once they're liquefied in the stomach, are going to clump up into a hydrophobic little droplet or ball. And it's not going to be very easy to get in and break those lipids down with enzymes. So emulsification in the digestive system is carried out by something called bile. Bile is made by the liver and secreted into the small intestine, where it mixes with the food coming out of the stomach. And then your stomach doesn't have anything to do with breaking lipids down. Lipids aren't going to be broken down until they get to the small intestine. And so you need that emulsifying agent of bile to get there. If you do not have anything to digest, the bile that the liver produces will get stored in the gallbladder until you've eaten a meal and you need a little burst of bile. That's all the gallbladder is. It's just a storage container for bile. And when we get to talking about the liver and the gallbladder, I'll talk about that a little bit more. Okay. But what I want to spend a little bit of time on here in the first half is talk about enzymes. Now, this is a table from the book that lists a number of different enzymes. Starting up here at the top with salivary enzymes, and then gastric enzymes. Salivary, obviously, in the saliva, gastric referring to the stomach. And then the next half of this table is populated by what are called brush border enzymes. The brush border is the apical surface of the small intestine epithelial cells. And we'll see a picture of that later on. But that's what's called the brush border. And there are enzymes associated with that to break nutrient molecules down to their last final stage before they get absorbed into the small intestine cells. And then after the brush border, which I would have put before the brush border in this list, but whatever, are the pancreatic enzymes, which are nearly as long a list as the brush border enzymes, maybe even longer. But the pancreas produces a bunch of enzymes that are secreted into the small intestine, and they help with breaking things down. Now, I would have put the pancreatic enzymes before the brush border enzymes because we're really sort of talking about the order of digestion. We start digesting some things in the oral cavity through salivary enzymes, and then other things get digested in the stomach, and then the pancreas provides most of the enzymes that break down larger macromolecules in the small intestines. And then what's left over from that enzymatic breakdown, the brush border enzymes take care of at the last step before it gets into the small intestine epithelial cells. Okay. Now, this is the list out of the book. It's in that last section of the chapter that's on chemical digestion and some other stuff. But I'm actually going to, for now, use this table. Now, I say for now because as soon as I get around to making the digestive system presentations that I'm going to put online, I need to take this out because this is out of a different textbook. But the table that's out of the book right now is not perfect. It's missing a few things. And I'd like to switch the pancreas brush border thing. So I have to manipulate the table and make it print out differently. But not right now. But this is essentially the same sort of list. The table is organized slightly differently. It's actually 3 different tables from the textbook that I got this out of. But they're organized based on which type of macromolecule they break down. So in the top we have carbohydrates, the middle is proteins, and at the bottom we have lipids and nucleic acids. Now, I want to talk about salivary amylase to begin with. This is one of the salivary enzymes, which is at the top of the list from the book, which is a good place to put it just as well. Any enzyme's name is going to end in ASE, and the rest of the word tells us what that enzyme breaks down. So amyl is referring to starch. So amylase breaks down starch. And it does that in salivary glands. So starch is a complex carbohydrate, and it's broken down into disaccharides and trisaccharides. Now, in truth, there might be some monosaccharides, okay? And that's an important point to make. So starch is basically a very complex chain of glucose molecules. Plants use starch to store glucose for when they need it. Where do we usually find starch in our food? Are those starchiest types of things that we can eat? Potatoes, what else? Pasta, those are the two big ones. Now, where does pasta come from, plant-wise? Probably most likely wheat. I mean, there are some like rice pastas and whatever, but wheat's a good example. And it's not just wheat in general, it's specifically the wheat berry, which is part of the seed that's going to grow a new wheat plant. And then where do we find potatoes? Where in the plant are potatoes? Anybody? People may need to drive up north sometime, take a look at all the potato plants up there. Right here in good old Pioneer Valley. Anybody? Really, you don't know where potatoes are on the plant? Well, they're in the soil, so they're part of the root, right? Now, how do you make new potatoes? Anybody watch The Martian by any chance? Movie, Matt Damon, stuck on Mars. Nobody? Great movie, go see it. He scienced the shit out of things. It's actually a line from the movie. So if you don't know the movie, Matt Damon plays an astronaut or Aresnaut, I guess you call him, who's part of a expedition to Mars. And just before, in the middle of their expedition, he gets a huge sandstorm comes along, which is actually a bunch of bull because that wouldn't actually happen on Mars. But looks good on the movie. And he gets hit by a flying piece of debris, and if the team doesn't get on the whatever spaceship, I can't remember what it's called now, to take off, then the sandstorm's going to blow the whole thing over, and they'll all be trapped on Mars. So they don't see what happens to him. His computer says that his vital signs have stopped functioning. They figure he's dead. They all take off. They save themselves. Turns out he wasn't actually dead. The thing that ran into him damaged his computer, so it reported that he's dead. So he's the only man on Mars, and he's got to figure out how to survive for, I can't remember how long it is, 4 years or something like that, till the next spaceship is scheduled to come to Mars. Nobody's seen this? God, it's really cool. Okay, go see it. You're gonna I know you're also dedicated to studying this semester, but as soon as you take that final, go see The Martian. Anyways, the reason I'm bringing this up is that he has to figure out how to survive for multiple years on Mars when they only packed enough food to last 6 people, 7 people, whatever it is, for 35 days or something like that. Now, since he's only one person, he can certainly extend the food out for a long time. But he figures out how to make more food because they also happen to be traveling up there about this time of year. And so they had a special Thanksgiving meal packed. So he took the potatoes out. See, here's where it gets back to class. Took the potatoes out and cut them up and planted those. Okay, you know, an eye of potato. Okay, you ever forgotten the potatoes are in the drawer and you come back later on, these green things running out of it? Those are new potato plants. Okay. The whole point of this, aside from telling you need to go see The Martian, is potatoes, just like wheat berries, are the starch storage location to help power new plant growth. Okay, so potatoes are going to grow into new potato plants. Potato eyes are going to grow into potato plants. Wheat berries are going to help the seed in the wheat grow into a new wheat molecule. And so that starch, that complex carbohydrate, is there and needs to be broken down. Now it says here that they're broken down into disaccharides and trisaccharides, which is mostly true. But actually, some of those complex carbohydrates will be broken down to release individual glucose molecules. What's an individual glucose molecule going to do if it's in your saliva? Think back to the beginning of the semester. glucose molecule in the saliva? What was the first topic we covered in this class? Senses, right? So what's a glucose molecule in your saliva going to do? What?
It tastes sweet.
Taste sweet, exactly, because it binds to a glucose receptor in a taste bud. Okay. Now, part of the reason why that works is because you're breaking down those complex carbohydrates. I've never actually been able to get this to work, but supposedly, if you put a piece of bread in your mouth and let it break down, eventually it'll start to taste very sweet. Okay. Never worked for me, it just starts to taste kind of bland after a while. But the idea is the amylase will break down the starch in there, and if you leave the starch in long enough, it'll break it down to disaccharides and trisaccharides, and maybe even break some of those down into individual glucose molecules, which will activate the sweet receptors. So if you're interested in trying this over the break, when you're having your dinner roll, leave it in your mouth for a little while and see if it starts to taste sweet. Of course, all of the butter that you put on kind of ruins the experience. Now, there is a pancreatic amylase also, and I'll just mention that because it's on the list here. But the pancreas makes an amylase that will continue breaking down complex carbohydrates in the small intestine. Okay. They start to be broken down in the oral cavity, but then they don't get reintroduced to enzymes until they get back down to the small intestine. Also in the small intestine, we have these enzymes that are part of the rush border, and they take specific disaccharides and break them down into monosaccharides. So let me get out of the presentation for a sec and go back to writing things down here. So there's maltose, sucrase, and lactase. And they're named simply for the disaccharide they break down. So maltase breaks down maltose. Where do we find maltose? You might be more familiar as it's normally just called malt. What's the source of malt? Think about a beverage that you make from it. Sorry? I was going, when you say malt beverage, you're usually referring to a beer type thing, but it's made usually from a grain, like wheat or something like that. So malt is usually from that. Now, malted milkshakes are having malt added to them to give them a different type of sweetness. That's all I can say about it, because it's a different mix of sugars. Okay. Now, sucrase is going to break down sucrose. Where do you find sucrose? Yeah, it's what we call table sugar. Okay. So whenever you sprinkle sugar on something, you're adding sucrose to it. Okay. Oh, I meant to say this. Maltose is a glucose molecule bound to another glucose molecule. Okay. Sucrose is a glucose molecule bound to a fructose molecule. Now, glucose is C6H12O6. We introduced it last time when I talked about the cellular respiration reaction. Fructose is C6H12O6. Okay. They have the exact same 24 atoms in them. The difference is just how those 24 atoms are arranged in the molecule. So glucose is a 6-sided ring structure with the extra cyclic carbon in the first position, I think. Fructose is a 5-sided ring structure with 2 extra cyclic carbons, one in the first and one in the 4th position, I think it is. Which is just to say they're differently shaped molecules, but they're still the same thing. Okay. If you break maltose down, you get 2 glucose molecules. If you break sucrose down, you get a glucose molecule and a fructose molecule, okay, in a 50-50 split. Now, when you hear fructose, you probably associate it with what? a common reference to fructose these days. Yeah, high fructose corn syrup. Now, what have you usually heard about high fructose corn syrup? Not good. Why? Okay, just not to pick on you personally, but if you hear that things are not good, you don't know why it's not necessarily good, just don't pay attention to it. Now, it might be true, but you need to have an idea of why, yeah, it's, playing with live firearms is not a good idea, but if you don't understand that the reason is that you could shoot yourself, whatever. I don't know where I'm going with that. But the point, actually, where I'm going with that is that high fructose corn syrup gets a lot of bad press. And a lot of it is more political than anything else.
Because it's cheaper now.
Sorry?
The only problem is that it's cheaper.
So, humans have an automatic response to sweet tasting food, which is to eat more food. Now, the reason for that is glucose is the only, or carbohydrates are the only fuel source that our brains can use. The rest of our body can use fats and amino acids, but in our brain, we leave fats and amino acids alone. So if we find a carbohydrate-rich food source, we want to eat more of it. We're hardwired, and when we taste something sweet, we eat more. Now, high fructose corn syrup is a cheaper source of sugar than sucrose. Where do we usually get sucrose from? Yeah, everybody's going to say that thanks to high school history books. But actually, that's less common than sugar beets. Sugar beets are the primary source of sucrose. Sugar cane can only be grown in very particular climates, so it's not as common and it's a little harder to farm. But there's a great YouTube video about sugar beets that has a woman standing in front of this huge pile of sugar beets. It's like 10 times taller than she is. And it's from whatever state sugar beets are common in. I can't remember now what it is. But anyways, when you get sucrose out of a sugar beet or sugar cane, if that's what you're going to get, then you're going to have a nice 50-50 mix of glucose and fructose. High fructose corn syrup obviously comes from corn. And corn is a lot easier to grow, a lot less expensive to grow. But you get a mixture of about 60% fructose and 40% glucose in that. Also, I believe high fructose corn syrup is all monosaccharides. So you don't need an enzyme to break it down. It's already sweet. So if you add high fructose corn syrup to food instead of sucrose, it's going to make the food taste sweeter, which would seem to say that you could make it taste sweeter more cheaply. A little bit of high fructose corn syrup should make something taste as sweet as something made with a larger supply of sucrose. Okay. That's not how big food thinks. Okay. Big food says, hey, if we make it taste sweet, they'll buy more of it. So let's make it taste really sweet so they buy a lot of it. And that's why high fructose corn syrup gets its bad rap. It's a very easy component of food to add in, and it's fairly cheap to grow and make. But instead of using it efficiently so that food's as sweet as it would be with a less expensive additive, the additive is there really to stimulate our inherent need to eat more when we taste something sweet. Okay. So it's not actually something about high fructose corn syrup itself that's bad. It's that it's misused, that it's there to make things taste sweeter, not necessarily more caloric, but it entices us to eat more food, which then means we take in more calories. So that brings us to lactase, which breaks down lactose. And lactose is a glucose molecule bound to a galactose molecule. Okay. So again, glucose is C6H12O6, fructose is C6H12O6, galactose is C6H12O6, different shape. I think with galactose, and I might be wrong because I'm not a biochemist, I think that it's a six-sided structure with the one extra cyclic carbon in the 4 position. Again, I might be wrong about that. but it's a different shaped molecule than glucose, slightly different. Where do we get lactose? Not lactose. Sorry. Where do we get lactose? Here's milk sugar. Now, we're mammals, and mammals are defined by the fact that we get our initial nutrition through milk sources from maternal milk. Humans are different than most mammals because we have dairy agriculture, which is we get nutrition from mother's milk, but not our mother's, a cow's mother, or a goat's mother, or a yak's mother, or whatever you happen to milk. We can potentially make something of that. Okay. So let me go back to the presentation here. Whoops, wrong. Okay. So I have this picture, which comes from a Nature article. Nature is one of the big scientific publications out there. And this particular article is Archaeology, the Milk Revolution. So this is actually a paper about archaeology. What they did is they unearthed pots in a number of different places, and they used biochemical tools to assess what was contained within those pots. And so what they were looking at actually were molecule evidence of dairy agriculture. And what they did in the paper is they combined that archaeology and biochemistry study with a more straight genomic or genetic study of the prevalence of lactose tolerance. Now, I said a little bit ago that humans are unusual because we have dairy agriculture and we take in dairy products for more than just our mothers. But really what it is that humans are unusual because we can take in dairy as adults. It's actually not the most common situation. We should, like all other mammals, stop being able to digest lactose as after we pass our newborn stage. However, there have been groups of or populations of humans that have developed a lactose tolerance into adulthood that apparently corresponds with dairy agriculture. So this picture from this archaeological study of pot shards that contain milk molecules in them showed hot spots where lactose tolerance was common. So we have here on the equator, far Western Africa, Saudi Peninsula, and Eastern Indian subcontinent, and then Northern Europe. Studying the genetics of a number of different people and tracing their lineage back, I guess ethnically or something, they came from those particular regions. So people today that have lactose tolerance, they can trace them back to coming from those particular ancestral lands. And these also correspond to where the people found pottery that had signs of dairy agriculture. Now the paper, which only came out a few years ago, 2013 actually, and I think it was early in 2013, so almost 3 years ago, was suggesting that the prevalence of the genetic marker for lactose tolerance corresponded with dairy agriculture. I think it was after this paper came out, I read a more updated study that talked about this, and the data that they were talking about was based on archaeological finds of dairy agriculture, I think 8,500 years ago. But genetics are showing that humans were only capable of dairy processing, lactose tolerance as adults, about 3,000 years ago. And so the interesting thing that came out of the later study was probably where there's dairy agriculture 8,500 years ago, lactose was broken down by other organisms, namely microorganisms, bacteria. So where there was common dairy agriculture 8,500 years ago, yogurt was an important staple of that, or cheese, where milk products were cultured with microorganisms that would make it more digestible for the humans to ingest. But with that prevalence of dairy agriculture making use of microorganisms to do the initial digestion of lactose, the tendency of people to develop lactose tolerance increased, and so that's why we see these hotspots. Here on the equator and in Northern Europe are places where we tend to see primary evidence of dairy agriculture. So it kind of fits with things there. The next part of the table, again from the old book, is talking about proteases. I just want to mention two things here. One is pepsin, which is made in the stomach, and I'll talk about that a little bit later on. In parentheses, they have pepsinogen, and up here at the label, they have enzyme, and then in parentheses, proenzyme. And there are a couple of other ones that are proenzymes. Those are things that are made by a cell and secreted, and then they're activated and become an enzyme. So these chief cells of the stomach make pepsinogen and secrete pepsinogen into the stomach. But then when it gets into the stomach, pepsinogen becomes pepsin, which can break down proteins. Now, the reason why you need to have something like that is you don't want the cells that are making the enzyme to be digested by the enzyme they make. So they make an inactive or proenzyme form to secrete, and then once it gets into the location where it's needed, it changes. For pepsinogen, what makes it change is a pH change. So inside the cell, pH is going to be fairly neutral, like we would expect. When pepsinogen is exposed to stomach acid, it changes into pepsin. The other thing I want to point out, since I was talking about dairy agriculture, is rennet. Now, there's two reasons I want to point this out. One is kind of minor, which is there are two molecules in this course that get mentioned that are both pronounced renin, and they're spelled differently. This one is renin, and this is an enzyme that's made in the stomach to break down milk proteins. The other one that we're going to meet is renin, which is a hormone that's made by the kidney. Now, the hormone made by the kidney really should be pronounced renin, but everybody pronounces it renin. But just keep in mind, renin, the root of that is rene, like renal. Renin is, its root isn't just rene. It's renin, whatever the root forms. Now, renin in the stomach, this enzyme that I'm talking about, is secreted only in infants. And its point is to break down milk proteins, because again, infants primarily are getting their what do we call that? Nutrition from mother's milk. Now, lactose tolerance is addressing the milk sugar. There's not a milk protein tolerance that we develop as adults thanks to dairy agriculture. Instead, some of these other protein enzymes can break down milk proteins. But renin is made in the stomach and plays a role in breaking down milk proteins there. Anybody ever made cheese? The fact that nobody really had made mayonnaise before, I doubt anybody's made cheese. But anybody? Anybody made cheese? By accident? I bet you've all made cheese by accident. Anybody forget to throw out the milk? You pour it out and it's like gross stuff. That's cheese. It's cottage cheese, technically. Add some salt to it, it's cottage cheese. Now, it's really disgusting cheese because what's happened is the proteins in milk have been partially digested. And what happens when you have cheese milk go bad is it's acids that's not breaking it down, but at least denaturing the proteins. When you're making cheese on purpose, you don't want the milk to spoil necessarily, although you might add some acid to help the process. Instead, in making cheese, you actually use renin as the primary coagulant, which it says down there in the end, causes protein coagulation, which is it breaks apart the milk proteins, and sort of like egg whites go from the clear runny stuff in a raw egg to the rubbery white stuff when you cook it. Milk proteins, when they're coagulated, will take on a different consistency. Does anybody remember the Little Miss Muffet rhyme? Anybody? Little Miss Muffet's out in her tuffet eating her curds and whey. Remember that one? That's the only part that's relevant here. Curds are coagulated milk protein. Whey is the water component of milk. Okay? So if you separate the curds and whey, you drain the water out of the spoiled milk. The curds that are left over are essentially cottage cheese. Okay? I think it might be referred to as farmer's cheese, but I don't know if that's exactly true. You add a little salt to it, and that's cottage cheese. Give it some flavor. And then if you wrap that up really tightly and put it in a cold, dark space where some microorganisms will enjoy it for a few months, when you come back to it later, you have hard cheese, cheddar, blue cheese, whatever it is. Depends on where you expose it, how long it's there. That's how you make cheese. Okay? And it comes from rennet. You need a little bit of actually calf stomach to start making cheese. That's the source of rennin to coagulate the milk, if you don't want to do it the bad way and spoil it all first. But you can do that. Nowadays, chymotrypsin is used primarily. I think it's chymotrypsin, which is made through a bacterium. I don't know if the bacterium necessarily makes chymotrypsin on itself or if it's been genetically modified to do so, but you can get chymotrypsin if you make cheese at home instead of having to use calf stomach extract, which might sound a little disgusting. Which also means if you use chymotrypsin, technically it's kosher cheese, but that's beside the point. Then we have lipids get broken down by a molecule called lipases. There are two listed here, and in our book they list them also. The pancreas makes a lipase that helps break down lipids when they get in the small intestine. And then they refer to lingual lipase, which obviously is made at the time. I don't believe in lingual lipase, which is to say, I don't think humans actually make lingual lipase. Most of the references I can find to lingual lipase are actually based on studies done in rats. And rats and humans, while we're very similar on a number of different physiological points, this is not one of them. Humans and rats are omnivores. We can eat meat or vegetable, get nutrition out of both. But we are hunter-gatherers. They are scavengers. Okay? So rats are just looking for any kind of caloric source. And so lipids have high calories, and they're a great source of food for rats. For humans, remember I said earlier that glucose is a very important molecule for brain energy, and so we're primarily looking for carbohydrate sources. So I don't think that we make lingual lipase in any appreciable amount because it doesn't really help anything happening in our oral cavity. When we talked about sensory systems, I introduced that there are 5 senses. A sense of lipids was not in there. It is possible in the future that it will be discovered that humans do have a sense for lipids, but I really doubt that'll happen. It has been established that we have genes that can code for lipid receptors, but there's no evidence that we express them in any taste buds in our tongue. So I don't think this really is true for humans. And it's more the lipids are broken down in the small intestine by pancreatic lipase. Another thing that supports this is emulsification. The only emulsification that happens in the mouth is a physical emulsification when we're chewing. And it's unlikely that that's really going to do the job because more likely it'll cause the hydrophobic components of our food to kind of gum together again in sort of lipid deposits. But when we get down to the small intestine where the pancreatic lipase has its job to play, the bile from the liver is present to help emulsify that so the enzymes can get in there and do their thing. And then finally, it's here I want to point out, nucleases are enzymes that break down nucleic acids. If you look on a nutrition label, you will see reference to carbohydrates, proteins, and lipids, fats. But you will not see any reference to nucleic acids on a nutrition label because nutritionally they aren't important. They don't have any real caloric sense to them. But all of the food that we eat ultimately comes from some sort of organismic source. And I use organismic instead of organic, but I want to tie it up into the political nature of what organic food is. But anything we eat ultimately comes from some sort of organism in the past. Something has made the substances that we're eating. Okay? Simplest example of that is an egg. Okay? Anybody have eggs for breakfast this morning?
Just okay.
So you had DNA in that egg. Okay, there's one cell in that egg that has DNA compared to all of the lipids and proteins and, well, there's not really carbohydrates in eggs, but all of those other things there that are there to support that one cell that's eventually going to become a chicken or whatever it might be. Now, it might be more than one cell, but primarily we should get eggs before they start developing into chicken embryos. So there shouldn't be a lot of cells in there, and therefore not a lot of nucleic acids. But also if you eat vegetable matter or meat, you're eating cells from other organisms, so there's going to be DNA in there. But the amount of DNA by weight is insignificant compared to the amount of DNA amount of these other molecules by weight. So we don't really consider it a nutrient molecule, but we do have enzymes that break down nucleic acids, so we can use those nucleotides to some degree when we need them. Brings us back to the overall list here. So that gets us to what I want to talk about in the first half. So let's go ahead and take a break, and we'll come back and I'll go through the functions from the oral cavity through kind of into the large intestine. And we'll finish out the day there. So about 20 minutes, be back in here by between 20 and 25 after. The sooner you're back in here, the sooner I get started, the sooner I finish, or at least the less repeatedly I have to speak at the end. Fast, quick, go ahead. Take a break. See you in about 20 minutes.